My name is Mark Band. I'm the director of the Functional Genomics Lab at the Carver Biotechnology Center. We specialize in genomics technologies, I would say all genomics technologies aside from sequencing. A lot of the technological developments, first of all, there are new processes, and there are processes that are on much higher throughput basis. One of the things that we're doing now, quantitative PCR, where we used to look at individual reactions in a 96-well plate for 96 or 384 reactions at a time. Today we have technologies that can look at multiple genes for multiple samples. So we have instruments that can do up to 9,200 reactions in 2 hours. So if a student or a postdoc thinks about pipetting out all of those reactions and setting all that up in their lab and running them plate by plate, it can take weeks. Today we can do that overnight. A lot of the research is either very focused on specific types of experiments. Others are done on a sort of a basic research initiative. Both are important for the development of science and the development of genomics. Some things can be very practical, like looking at the microbial development in the gut of pediatric patients that are either fed on mother's milk, breast milk, as opposed to formula. Seeing what types of changes in the gut are happening. So this would be on a much more focused type of level. Other things are looking at just screening different types of conditions. So you may be looking at soil samples from different types of soil under different fertilizer regimens, under different agricultural processes, things like that, and building up a database of what's there. This can then be used for more focused types of research. The technologies in our labs, particularly in the Functional Genomics Lab and the sequencing labs, are able today to analyze many different types of organisms, animals and plants, which have not been classical model organisms for biomedical research or agricultural research. Today we run samples on honeybees, bumblebees for neuroscience, stickleback fish, also for behavioral analyses. I have a collaborative grant with another group on mole rats, which are used for studying hypoxia tolerance. As an underground animal, they can live in very, very low levels of oxygen. And other types of animals, there are birds that live in the Andes at different elevations, the same types of species. They can be used to look at evolutionary development as a hypoxia model. And of course, human beings today are the biggest model or most useful model organism that we're using. On this campus, more information is better in general. There are problems with handling all this information, so you need new algorithms to analyze these types of things. But the more data you have, the more coverage you can have. In other words, if you're looking at 5 million different regions of a genome, you have a much better chance of focusing in on a particular region that actually influences whatever you're looking at, if it's health, disease, phenotypic traits, things like that. And I think we have a very interesting collaboration on campus because the campus is a very well-renowned computer science and engineering, so the bioinformatics has made great leaps on this campus, and it integrates very well with the high throughput technologies that we're using in our labs. So we have both research collaborations and basically contracting services. We do have collaborations with researchers at the local hospital, Carle Hospital, in the oncology department, looking at tumor cells and circulating tumor cells on an individual cell basis, looking for biomarkers that may give hints at what types of cells may metastasize. We have collaborations with other researchers in many other different universities both through collaborative grants and just people sending samples to process here. I think just because we have a very good name in terms of the quality of the data that's produced in our labs, many researchers from well-known universities that have sequencing centers in the same types of labs in their core facilities will prefer to send here just because of the data quality that they're able to get from our labs.